Okay, come. People. There are multiple topics I need to go through today, so however, I'll only focus on the main things. I'll show you which one that you will need to take note of, alright? So the first one I want to go through is your cell division. Typically for cell division, number one, they like to ask, okay, is this structures of your cell division. So let's go through the basic structures first. First one, which is only present in animal cell, this is always your centrioles here. Now, however, again, for your centrioles, I want to talk about this. This is actually can be found on your first page of your notes, okay, your second column. In exams, they always like to ask, identify this particular structure of your centrioles and then give evidence to showcase. There is always two key phrases we always describe for your centrioles. Mani, what's the first one that we always use to describe centrioles there? It's always a, so for those who just received, make sure you write this in your notes up. A pair of rod-like structure. That is, just now you want to mention?
There is
perpendicular region. Yes, a pair of rod-like structures perpendicular to each other is the first, okay, distinguishing feature. Second distinguishing feature is what? Any idea, Chloe? It's always made out of how many microtubules? 3 by 3 by 3. How many? 27. No? Oh, yes, but 9 triplets. The way we write, don't write 27 microtubules. You say 9 triplets of microtubules, alright, arranged in a ring. This is always the description because in exam, this is how you actually typically write for exam, yeah? Okay, now moving on. What is therefore the role of your centrioles? I don't think I went through this for you. If they ever ask you what is the role of centrioles, there are always, again, two major roles you write for centrioles. I know the phrasing, right, it is actually very crucial. One of the phrasing we write for the role of centrioles is centrioles actually, number one, produces your spindle fibers. When you want to tell me how it produces spindle fibers, okay, at your microtubule organizing complex, alright? So this centrioles produce spindle fibers at the opposite poles of the cell towards the equator of my cell. From opposite, sorry, my bad. Opposite poles of the cell to equator. This is called produce spindle fibers because the other is actually organize your spindle fibers. So they organize the synthesis of spindle fibers. If you want to mention this, they are all different. They sound the same, but they are not the same. So the other one, organized synthesis of spindle fibers, which lead to the equal separation of sister chromatids. Alright, so if they ever ask you what is the structure features of your centrioles, always remember this too is not the first time you all should be seeing this. It's always repetitive, understand? Alright, rows is down there. The second thing we need to take note of is this centromeres. Please take note, centromeres are non-coding. But centromeres have, again, usually when we want to describe centromeres, which is this, your centromeres, we always say, is a non-coding region of tandem repeats. This is the typical phrase we describe, okay? So it consists of non-coding DNA made out of a series of tandem repeats. Tandem repeats are like your repeating sequence. Hopefully you know it's AT reach. Another thing you need to take note about your centromeres is that centromeres are heavily methylated as well, okay? So this is something that we need to take note of. Now centromeres, they are actually associated with two types of proteins. For exam, the number one protein you always think of is what? Any idea, Megan? Kinetochore. So one thing is that you should know your centromeres is complementary in shape. to your kinetochore proteins. But if you ever want to mention, it is actually going to associate you on these kinetochore proteins. How do you actually write for exam? You cannot just stop there, right, Megan? So again, it's complementary in shape to the kinetochore proteins. Okay, to bind. Why? Don't stop there. What must we further elaborate so I can get 1 mark? For my girl. So you must write to attach. A lot of times if you never write to attach, right, you realize you never get marks. You must say to attach. Now, we learn, right? There are 2 reasons, right? Let's see, Satu do what? First reason, we need microtubules to attach. And I want to hear the keyword they typically write for exam. So what's the first one, dear? Ensure equal separation is the keyword of sister chromatids, or you can say chromosomes if it's meiosis, to opposite poles of the cell. Keyword, I repeat again, is equal separation. Don't write opposite sides, only poles, north pole, south pole. Myra, what's the second thing? I give you a hint, it's under your metaphase. To ensure, keyword is what? Keyword is called proper alignment. Alright? So again, I need you to take note. This is not going to be the first time you see the word proper alignment. So proper alignment of chromosomes along the metaphase plate or equator. Alright? So you need kinetochore proteins to actually attach. However, I said that in syllabus, I taught you another protein so you will know. In exam number 1, right, this is always the number 1 function you will write for your centromere. Understand? So this is where your kinetochore protein actually assembles and the microtubules will actually from each centromere attach to it. But there's another set of protein that will actually bind to. What is this one that I'm trying to label in orange? Any idea, Colanthia? That also allows sister chromatids to combine. So it will be complementary in shape and charge to your this other protein. Any idea? No? You? Very good. So this is actually complementary in shape and charge to another new protein that I will teach you. It's called cohesin protein. Sorry. So again, your this cohesin protein, okay, will bind. The purpose of this, don't stop there, for sister chromatids to adhere to each other or new to each other. Okay, but what is the reason we write for exam? How come your sister chromatids must adhere to each other, Vivian? Because to ensure no proper. Yeah, if they are not adhere to each other, you cannot align them equally or in a single file or double files along the metaphase plate. Whether is it mitosis or meiosis. And then again, what happens to your this cohesin proteins during anaphase? So you can separate them equally. During anaphase, you will be cleaved. So cohesin proteins are usually cleaved or degraded, okay, during anaphase, and this will ensure equal separation. So do you see the terms proper alignment? Equal separation is always the common association of phrases. Okay, so take note about this. Here, so you have your this centrioles, you have your this proteins, but we also learned that your this cell division, we need to also learn our this normal how cell division is actually regulated in your typical cell, right? So let's see whether you recall. Cell division, right, we know it is well regulated because we have this thing called checkpoints, right? What are my 3 checkpoints there? Okay. For your G1, G2 and M checkpoint, G1 and G2 are part of which phase of cell division? Interphase. And then we know that this M stands for metaphase, right? Agree? So let's do a quick recap. I said before, let's say if you cannot remember what do they exactly do, alright, which I still later go through, we still need to learn their overall two functions, right? Why we need to have type control, right? So, why? PPP. So the first one, I always say is PPP. PPP stands for what? Prevent premature progression of the cell cycle. Or another phrase, if you cannot remember, is just to prevent excessive cell cycle progression. Or you can say prevent uncontrollable cell division, which you know that if it actually occurs, it leads to cancer. Okay? What else do we also need to take note of? What is the next function? Any idea, Myra? Why do we need to have all these checkpoints besides prevent premature progression of the cell cycle? Is to provide, let's say there's DNA damage, what must I do? Yes, so we need to also provide sufficient time, okay, for cell machinery to repair itself should there be any damage. Okay, now for G1, G2, each one of them has their own. This is actually relevant and this is essay. So let's still go through. So we learned that your G1, this is right before your S phase, which is your synthesis, also known as when semi-conservative DNA replication will occur. You always know at this particular point we need to check if cell size is adequate. But most importantly, we need to ensure two other things. What are two other things we need to ensure before we can undergo semi-conservative DNA replication, Kalentia? Like besides checking cell size is adequate, what must we take note of?
Enough nutrients.
Enough nutrients, sure. Sufficient nutrients under here. But I'm actually looking out for whether there is presence of growth hormone. Why? You all should know already. Cell division, okay? Cell division, cell division is not going to be a chapter that they like to test as a topic on its own. It's very rare. Sometimes you can interlay and overlap other topics together. Some topics they like to overlap is like your this mutation, cancer, stem cells, okay, or even cell signaling. You should know already, there must always be signals to instigate your this cell or stimulate the cell to divide. So that particular is called your this growth hormone, which is actually your ligand. Understand? To stimulate. So in order to make sure that it doesn't have dysregulation of checkpoints, they need to see that, there's actually a growth hormone, there's a proper channel to actually stimulate cell division. Understand? Another one, you always need to check for what? DNA damage. Because it's right before your this S phase. So that's why if you have actually any DNA damage, you should know already, this will actually pause, repair the damage before they can proceed. Okay, clear? Then S phase, you have semi-conservative DNA replication, a quick one, Satu Dua. What is semi-conservative DNA replication, dear? First one? So both parental strands act as a template. What's the second one, Kalentia?
The cell is made up of one old parent strand and one male.
Yes, understand? So take note. Clear? Next one, G2. G2 is that, what do we check for? We are trying to ensure DNA replication is complete. This is the main thing we need to take note of. Obviously, we also want to check there's sufficient organelles. As well, okay, before we can undergo mitosis or meiosis. Alright? And checkpoints. So, Okay, this is actually obviously to ensure proper alignment. Okay, kinetochore microtubules are actually attached. I need to emphasize on the word kinetochore microtubules because we learned that such thing as polar microtubules, right? Polar microtubules do not attach to the kinetochore proteins. What's then the function of polar microtubules? Wait. Wait. To elongate your cell. Understand? Because they will overlap to elongate your cell. So at centromere, alright, from both poles, and then again we're trying to ensure there's bipolar tension. Okay? From both sides. Here. So learn why do we have all these checkpoints. Ready? Now, but one thing we need to take note of is, again, for your this cell division, I'm not going to go through all the basic things of what happens during the phases of mitosis, meiosis. That one you're going to study yourself. You all should know one main difference between mitosis and meiosis already. This is actually on the last page of your notes. Okay, is that mitosis, there is again only one cell division. Okay, just to separate your sister chromatids. Your meiosis, you have two cell division, but the meiosis one is actually to increase your GV. But if you can flip onto the second last page of your notes where you see the heading called importance of meiosis. I need to point out about this. Because importance of meiosis, there are two main importance, and then later the second importance we need to learn why. Do you see the first importance of meiosis I have is for sexual reproduction? My question is, meiosis needs to form what kind of gametes, Chloe? Haploid gametes. Why the need to form haploid gametes? In order to restore diploid condition upon fusion of gametes. This is always the standard key phrase you write down at your this sexual reproduction down there, the heading. This is on your this, if I'm not wrong, your first column on the second last page of your notes. Okay, so again, you will say is to form haploid gametes in order to restore diploid condition upon fusion of gametes. Okay, so I repeat, to form haploid gametes in order to restore diploid conditions upon fusion of gametes. But let's ask. So the main thing I want to ask is your this cell division. Cell division, right, like meiosis, also have another one to increase GV. But what's the importance of increasing GV first? What do we typically write for exam? Increase GV for what purpose, dear? Of a keyword? Especially during changes in environmental conditions. I'm emphasizing on species because some students always like to write increase GV in an organism. You can't increase and have all different variation in a single organism. It's changes in a species. So again, the reason why we always need to know the importance of your GV is actually you write increased GV will increase chances of survival. This is very related to your natural selection. Chances of survival of a species, especially during changes in environmental conditions. But let's do as a recap right now. Those that increase GV, there are actually three things that help to contribute to GV. And then you will be explaining to me how they increase GV. In my notes, I only have two, but I want to emphasize all three. Ready? Myra, can you tell me what is the first one that contributes to GV during meiosis? And there is always the standard phrase that we write. It's called what? Between of. So you always write between, not haven't finished yet. Non-sister chromatids of homologous chromosome during one. Yes. Okay. Of homologous chromosomes. During your this prophase 1. So your this crossing over, you hopefully know, under inheritance is testing on what kind of linkage, Kalentia? What kind of linkage is it testing for crossing over? Not sure? You? What kind of linkage? Sorry? Your head. This is partial linkage or incomplete linkage. Now, what do I mean? Did I erase? So, typical generic diagram if you want to draw crossing over is this. But remember your this, not sex linkage, is your this partial linkage. So for example, we learned they are linked on the same chromosome. Imagine you're this one, small a, small b, small a, small b. If I am crossing over, do you agree here? I just focus on this one. Do you think here, potentially can I now have a big A, small b combination, small a, big B combination? This is the gametes that I can form, right? And then this one, can I also for this do you see how this part here, I can have a big A, small b combination? So, previously these are only gametes that I can form, right? So, however, do you see now I form new combination of gametes? New combination of alleles. So, we learned that this contributes to GV, right? At the site, how does this actually increase GV? How do you write in an exam? This will increase what? Yes, so the way you write how it increases GV, very different from contribute to GV. This contributes to GV, but how it increases GV? It creates new combinations of alleles is the phrase. Do you see this new combination of allele I formed here? And then that's why I can give rise to that. There's a second contributing factor of GV. Wani, what is it?
Independent assortment between
Of what?
homologous chromosomes
During?
during metaphase 1.
Okay. Now, during metaphase 1, if I draw, we always have combination of maternal and paternal chromosome. But do you agree, random assortment, right? It doesn't mean all the maternal chromosome must always face one side. All the paternal chromosome must face the other side. Sometimes it's just random assortment. Wani, can you tell me, how does this actually increase GV? Two ways. Number one is what?
Creates new combinations between the paternal and maternal alleles.
So again, I want to see is it creates new combinations. This is very different from that. But your new combinations of maternal and paternal chromosomes, not alleles. Know the phrase well. What's the second one? It also can increase it by, how does it increase GV, Chloe? It creates how many possible gametes? That's why even though crossing over is very rare, you still will not look exactly the same if it does not occur. 2 to the power of what? Hello, my Sec 4 student also can tell me. 2 to the power of n. You know what n represent? What? Your head No, you tell me right now, for humans, how many possible gametes combination you can form? 2 to the power of what? N represents haploid number. So you can form 2 to the power of what? You got 32 chromosomes. What's your haploid? Okay, how many chromosomes you have? Can you tell me? You have 64 chromosomes? I want to study your body. Hello, you only have 46 chromosomes, dear. 46, you only 46, So what's your haploid number if your diploid number is 46? So how many possible gametes you can form? 2 to the power. This is a very large number, right? So please take note, independent assortment, you can create 2 to the power possible, 2 to the power n possible combinations of gametes. Understand? Clear? Okay, third one. So number 3, there's also another one. Can you tell me what's the third contributing factor? Random fusion? Oh, random fusion? Random fusion is not part of meiosis. I'm talking about meiosis. The metaphase 2. Give you an example, metaphase 2. What happens during metaphase 2? Random? Okay, question. By the time you reach metaphase 2, do you think some of the sister chromatids are identical? No, right? Agree? And then some more, some of them you may actually contain, okay, those that undergo crossing over already. But do you agree how each of your these non-identical sister chromatids phase 1 pole of the cell is random? So imagine now when this one I get 2, right? But some of your these sister chromatids are not identical to each other already. So what is this? Random orientation of non-identical sister chromatids. Okay, so this is during your metaphase 2. Okay? There's always a phrase, so the idea is how each sister chromatids, okay, that will face different poles of the cell may actually differ, not always the same. Alright? Clear? Something like that. So hopefully you all can see. Okay? So what I actually have, again, that's why so many different ways to form and increase PV. Learn. So hopefully you know. Alright, next one. So the next thing is, how come they need to test on this? How come they always like relate cell division with other chapters like mutation? Because everything we go through here, you need to think about possible ways you can actually form a mutation. We have 2 types of mutation. Alright, ready? I'm going to emphasize on these 2 types of mutation. So you can see, start to see it's actually related to your cell division. I'm going to erase enough. Enough? Now, we have 2 types of cell, the mutation that we have. I will just go through the basic first before I combine the similarities and differences between the gene mutation. Now, chromosomal mutation and gene mutation, they are not the same things. Now, I need to point out here, for gene mutation, never ever tell me, oh, nonsense mutation, missense mutation, or even silent mutation is a type of gene mutation. No. The type of gene mutation are your this substitution, your missense, as well as your this substitution, deletion, as well as my this what's that one? Insertion mutation. Understand? So the idea is, these are the types of gene mutation. But silent, missense, and nonsense are the consequences of this gene mutation. Never ever say that missense is a gene mutation. That's why we always say substitution mutation that led to what kind? Maybe silent, maybe missense, maybe nonsense. I know there's also another one called inversion, but usually these are the major 3. Now, so we learned that again for your this substitution, deletion, and insertion. Let's start off with the first one. Let's learn some of the causes. If let's say mutation does occur, which part of cell division this most likely, okay, might take place that can bring rise to gene mutation, Wendy?
DNA replication.
Which is actually what phase? So let's say this is most likely during your S phase. Do you see how it's related to cell division? So not only that, we also need to emphasize, okay, during which what could be a possible causes of your this mutation. So possible causes of all these different type of mutation before we go through in detail. If let's say if your substitution mutation is due to what? Errors of complementary base pairing. So that's why we can relate. Due to errors of complementary base pairing, okay, by DNA polymerase, that you can actually incorporate an incorrect nucleotide that can lead to substitution mutation. Agree? I also can have, again, things like, let's say your DNA polymerase, so errors in your this DNA polymerase, okay, that has mutation that incorrectly inserts a nucleotide. And then we know that this, again, what else can we also have? This one, your DNA polymerase incorrectly skips a nucleotide. But we learned DNA polymerase have proofreading, right? So what could be another possible cause, Wendy?
Can I say no free ribosome?
No, if you think about this, DNA polymerase is supposed to have proofreading.
Failure in proofreading.
So the idea, yeah, some part, you say errors in proofreading. Also can. By DNA polymerase. There are many ways that you can have all this mutation. So let's go through right now. We know that results of mutation is going to be your silent, your nonsense, and your missense mutation. Which one is most likely going to give rise to silent mutation? Can you tell me, Chloe? Is it substitution, deletion, or insertion? So substitution mutation can always give rise to what we call silent mutation. Let's hear what is the key phrase that is always linked to silent mutation. How come substitution can lead to silence? 1, 2, 3. First one, what do you say? Because of the what? Genetic code. Keyword is what? Story D. This is always linked to silent mutation. Degeneracy of the no variation. Degeneracy of the genetic. Again, is what? What does that mean, Megan? Later you write down tell me all the three to store that in your head. So it's what? Second one, we're the same amino. Or you can say the different codon still codes for the same amino acid due to what effect? Wobble base effect means what? Nucleo. Yes. Okay, what is the silent mutation? Due to what? Degeneracy is the first keyword. Second one is what? The different codon. Due to wobble what effect? Wobble? You never discover, you cannot come up with your own name. So it's wobble base effect. So due to wobble base effect means what dear? Change in the third amino or third nucleotide of the code will code for. Yes. Understand? Next one. All this, okay, can lead to missense mutation. Deletion also can lead to missense. Insertion can lead to missense. What is missense mutation, Kalantia? Missense mutation means what? Missense mutation means what? That means you change, when you change your nucleotide, Is it
It changes the
amino acid? So the thing is, here, can I now ask? I'm going to use this question this time to ask. So question is, substitution, deletion, and insertion mutation can give rise to missense mutation. So missense mutation means you change your amino acid. But which one can you tell me, Kalantia? Is it substitution, deletion, or again insertion? Will a non-functional mutation or non-functional protein due to missense mutation is more likely to occur? So I repeat, substitution, deletion, insertion. Which of this can give rise to a non-functional mutation to a higher extent?
insertion.
No. So, insertion only? That's the thing. You cannot just say insertion only. Also, deletion. Now, please take note. Insertion and this deletion, okay, can always give rise to a non-functional protein rather than a base pair substitution mutation. Why? Can I ask? Dear, can you tell me deletion and insertion, right? What's the phrase that we know they always lead to called frame something? This is going to be the key phrase that always linked to this, by the way. There is always a key phrase that's linked to frame shift. You all ready? Let's see whether you recall. Vivian, you recall? The reading frame is altered such that nucleotides. What's the key phrase?
Move by one.
Nucleotides downstream are then improperly grouped into. Never mind. Can you help me write this? Anything, this frame shift mutation under your notes, right, is on your second page of your notes where you see actually this thing called frame shift mutation. Can I borrow? So your frame shift mutation is located here, okay, on your this first column of your notes. Alright? From your this particular frame shift mutation, from now onwards, this is how you every time describe your frame shift mutation. That's why you can have this. Number one, first bullet point we always say is that when you have frame shift mutation, nucleotides downstream from mutation, that means either deletion or insertion mutation. Nucleotides downstream from mutation are keyword improperly grouped into codons. I repeat, improperly grouped into codons. Nucleotides downstream from mutation are improperly grouped into codons, your first bullet point. Second bullet point, how do we describe? Second bullet point, you always say, resulting in extensive missense mutation. Resulting in extensive missense mutation. That greatly alter amino acid sequences in the polypeptide chain and hence 3D conformation of protein. So I repeat, so you alter amino acid sequence in the polypeptide chain and hence 3D conformation of protein. You know that the moment you change your 3D conformation, okay, extensively, your this function of protein most likely is going to decrease, or actually no more function, non-functional. Understand? Okay, so know what is the phrase. Deletion and insertion and higher likelihood of your this missense mutation that forms non-functional protein. But just now I also mentioned, right, I'm doing this a recap, see whether you know. Very important for you to know why. Substitution mutation is less likely to form non-functional mutation despite having missense mutation. Can you suggest 1, 2, 3 why? So I repeat, very important. Substitution mutation is less likelihood of undergoing your this formation of your this non-functional protein despite the missense mutation. What is one reason why? I'm talking about missense first. Let's say you have changed. You know what missense means? Means you change the entire amino acid. What? Agree? But protein, why not I help you? The shape of your this protein is dependent on forming what kind of interactions? I cannot hear you. Sorry, is it? Only hydrophobic? You mean there is no hydrogen bonding? There is no ionic bonding? There is no disulfide interactions? So all these interactions are called what? R group interactions. Do you know what R group interactions are? Amino acids are different R groups, right? So your different R groups sometimes, do you think I only have one type of R group? So how many type of R groups do I have? How many types of R groups do you have? Oh, this is your basic, you all must know, under your biological molecules. First one, you can have non-polar R groups. We have your this polar R groups, and we have those that are charged R groups. Only non-polar can form your this hydrophobic interaction. Polar can form your this hydrogen bonds. This one can form your ionic bonds. Okay, only cysteine can form disulfide interaction. The question is, there are different type of R groups. Okay, my question is, do you think each type of R group only got one amino acid? This one may have more than one, right? This one have more than one, right? This one have more than one, right? Let's say I want to change my this amino acids due to missense substitution mutation. The amino acid change may have the same. When you have the same R group, can you form the same or similar R group interaction? Will it fold similarly? Will the conformation of proteins be almost the same? Will the function remain almost intact? That's the idea. So another reason why substitution mutation is less likelihood of forming a non-functional protein is because, number one, it may result in missense, okay? But amino acid substituted has similar R group properties. Amino acid substituted has similar R group properties. So what does this mean? Can form similar R group interactions. Protein conformation remains almost the same. Like you replace, let's say, glycine, okay, with another amino acid called valine. Valine and glycine all non-polar. Means they can still form, in your chem terms is your ID still what? So no change what? Megan, what's the second one despite missense? That means you change, so it's what, Megan? I do missense first, So let's say I did change my amino acid. Besides this property, this factor, what else? Maybe the amino acid you change is not found in the critical region of the protein. What do I mean? It's a non-essential amino acid. I give you an example. Enzyme is a protein, right? Maybe I change the amino acid that does not make up the active site shape. It's just outside the active site. So do you think that this enzyme can still bind to substrate to form enzyme-substrate complexes? Can. So a second reason is the substitution mutation may result in missense, but amino acid substituted, either you tell me it's a non-essential amino acid, or you say it's an amino acid not found in the critical region of the protein. Amino acid not found in the critical region of the protein. So again, protein function remains intact. Does it make sense? Makes sense. You, Myra, what's another one besides missense that substitution can give rise to still a functional protein? The famous one that just now I made her recite the full thing. The way you write, how come can still form functional protein despite silent? Because of the keywords of the, so the substituted nucleotide or codon still codes for the, so that's why you're this, due to, yeah, so that's why again protein's function or structure still remain the same. There's also another way we can substitute, okay? Let's say a substitution mutation occur, but yet you still form a functional protein. Any idea, Valentia? What else can I also give rise from substitution mutation, but yet I still form the same shape or function of the protein? Maybe mutation may occur where? Must it always be in the coding region? What could be a non-coding region? Introns. What happens to introns even though it's part of your gene? Yes. So this is also another mutation. Okay? So again, mutation can occur in introns. But what happens to introns? Introns are spliced or excised, okay, to form the mature mRNA. So that's why your amino acid sequence in your polypeptide chain remains intact. Your conformation of protein and the function of protein remains intact. So do you see how there are many ways they can test you? I'm just trying to jog your memory. Huh? Okay, causes of this, we also know for this chromosomal mutation, there are two types. First type chromosomal mutation is aneuploidy. It's chromosomal mutation in a number. Second type of chromosomal mutation is in your structure. Hopefully you do know aneuploidy is always due to one effect called what? Vivian? Non-disjunction means failure of sister chromatids to separate, failure of chromosomes to separate is just failure to separate. Structural mutation, again, is also when you have changes in your segment of this protein. If let's say I want to relate to cell division, when will this most likely occur? During what phase?
Interphase.
During interphase that you will have again non-disjunction. Prophase what?
1.
Most likely this must happen during prophase 1, your head. Anaphase 1 or metaphase 1 or anaphase 2 or metaphase 2. If I use again this mitosis, you will know it's either metaphase or anaphase. You know why? Failure to separate what? How can it be prophase 1? You're not even separating at that stage, right? So now let's see. We already learned it's due to non-disjunction. Let's learn the causes. So causes of aneuploidy. Let's try, starting with you. This we already know is failure to separate, right? What could have resulted in the failure of sister chromatids or chromosomes to separate equally? By right, just now, what do we say must have been attached? Must attach to where? What is this thing called? This is centromere. This is centromere. The protein. You must know this protein. It's like K. Yes. So again, sometimes, do you think, I say there are many ways, right? Elaborate, expand your thoughts. You're only learning a subset of many ways it can occur. So one cause can be what? Improper attachment of your microtubules to kinetochore. What can it also be? Like for example, I also can have cohesin proteins because this is kinetochore proteins, right? Cohesin proteins, what could be a problem? Yes, by right they're supposed to degrade or get cleaved, okay, during anaphase. But imagine if they did not degrade, they are not cleaved, wouldn't that lead to non-disjunction? Another one is, I can also have gene mutation. So I can have mutation of what? Centromere. When you have mutation in centromere, will it change the shape? Idea is that it can be no longer complementary to either kinetochore or your cohesin proteins. And that could also lead to non-disjunction. What? Okay, what else can I also have? There are many ways. Think about what else in cell division. Maybe you have mutation in your centrioles. Centrioles is supposed to form this spindle fibers. Example, for mutation in centrioles, maybe you have multiple, okay, mitoid spindle complex. Mitoid spindle apparatus. Imagine you have so many centrioles because of mutation. Well, then I form again, problems of non-disjunction. I'm just showing you some subset because sometimes they can suggest your brain cannot be so strict. Your brain must know how to interlink with other topics. Okay, structural mutation. If structural mutation does occur, this is most likely going to occur during prophase 1. Because you want to lead to your deletion, your insertion, your duplication, your inversion mutation, your translocation mutation. So, in other words, there are many causes. First cause, it can be, for example, you have misalignment. All this are, by the way, structural mutation usually is due to unequal crossing over. So how can you have unequal crossing over? So for example, it can be misalignment of homologous chromosome. Homologous chromosome typically must align in a single file along the metaphase peak. If they align incorrectly, they never go side by side, that means gene loci to the other respective gene loci, it will definitely lead to unequal crossing over. Agree? Second one, we can also have crossing over between non-homologous chromosomes. That's why it can lead to translocation mutation. Non-homologous chromosomes can incorrectly pair up and they undergo crossing over. Okay? Things like that. Many other ways. So know what's the difference between gene mutation and chromosomal mutation. Clear? But the question is, can I say chromosomal mutation will always have a less likelihood of bringing about, again, changes in your polypeptide chain? Sometimes, right, your nucleotide sequence may remain unchanged, but you should know, right, it also can affect your this chromosomal mutation. Like for example, what if it's, let's say, an inversion, okay, mutation. So I repeat, my question was, I know most of the time you all say, and we learned that yes, they will actually change, okay, something, but this one will change your polypeptide chain to a greater extent, right? It's true, right? Agree? Gene mutation. But can I say that chromosomal mutation, let's say structural chromosomal mutation, okay, can it always have this protein structure remain intact? No. You know why? There's a such thing as inversion chromosomal mutation. When you have inversion structural chromosomal mutation, in your notes you all know what it is. If I'm not wrong, it's on the 4th page of your notes, where you see the inversion one, where you swap. So you think about this. If I swap, this is my chromosome, by this region here, I'll just shape in here. Let's say at first one of your gene loci here, this is the 5' end to 3' end of the template. But when I undergo this chromosomal inversion mutation, do you see now instead of 5' reading frame, the 3' is here? The nucleotide sequence on the genes are all going to be read in the reverse manner. Will I have different codons being coded and your amino acid sequence is going to change? Yes. So that's why it also can change your polypeptide chain. It's just most of the mutation just change the level of expression of genes. Understand? So later we'll see, for example, like cancer is a famous one. But now I want to talk about similarities and differences between gene mutation and chromosomal mutation. So you can take note. Can I erase? Let's talk about similarities and differences. Ready? Let's see similarities first. Similarity number 1. What's the first similarity that I can actually state? Megan? question. Will both actually involve changes in your DNA nucleotide sequences? So if you cannot say yes, potentially can they at least change genotype? Yes. Okay. So both, okay, involve changes or alterations in DNA sequences. Okay, or you can say genotype, I also accept. I write in alternating color so you know things that change. Another similarity that I can also accept is that, do you agree? If you have all this, they potentially can cause disease. They also can cause abnormalities and even cancer. Think about your sickle cell anemia. For mutation, you should know some of the structural mutation is like what? Your this Cry-Ducat syndrome, your this chronic megalogenic leukemia, right? Your aneuploidy, Down syndrome. All this is also disease, right? Okay. I will not accept if you just purely tell me negative effects. You usually need to specify why is it bad. Okay, next one. This one, both can also change your this proteins produced or even your phenotypes. Another one, I can say that both also increase variation in the population. And that is also under what chapter? Evolution. Next one. What else? Again, we have is they both can be inherited by offspring. Alright, both you also can see can arise spontaneously and randomly. Causes of all this can also be due to radiation or exposure to certain mutagen like mustard gas. Okay, let's talk about the differences then. You should know now, first difference between your gene mutation and chromosomal mutation is gene mutation alters your sequence of your nucleotides on DNA. Whereas this one, okay, you will alter the structure or large segment of chromosome or number of chromosome. Idea is nucleotide sequence sometimes is mostly unchanged. How about which one has a change in chromosome number? You should know already. Gene mutation, oh I forgot to mention. When I say here, forgot to write N chromosome number and structure. Okay, again, you should know this will definitely change your this does not change your this number of chromosome. Whereas here, you should know it may result in change in numbers from aneuploidy. Or, for example, not necessarily must be aneuploidy. Polyploidy is also another effect. Think about how the fusion of gametes, okay, due to errors in fusion of gametes. Okay, so another one, please take note, this only affects only one gene, or you can say one gene locus, sorry. Whereas here, it affects multiple gene loci. Okay? Again, we also can talk about what they can result in. This can change. Okay? Your this phenotype. So let me just write here. It creates new alleles. Okay, so but this one, it only generates new combination of alleles on chromosomes, or it does not even generate new types of alleles. Okay, another one, we can say how this one can never be observed on your karyotypes, or the other one can. Karyotypes are this diagram will showcase every single chromosome a person possesses. Okay, these are the main things we just need to focus on. Now, why do we need to learn gene mutation and chromosomal mutation? Because mutation must occur and occur in accumulation, okay, in order to give rise to disease like cancer. So this is the first one that we need to take note of. Alright, so another thing, let's relate to cancer. I'm not going to talk about what is the difference between cancer cells and normal cells. You all can go and study that yourself. Okay, what I actually like to focus on is, again, what are the typical phrases that we learn from tumor formation and cancer formation. Again, what are some mutations that can occur that give rise to this cancer, development of cancer as well. Okay, so this is what I want to go through. Let's start off with, again, this mutation. Where is it? Okay, never mind. There's a difference between tumor and cancer cells. Not all tumors can become cancerous, but we learn that whenever we see a question, they say development of tumor, okay, or development of cancer, the phrase will always apply. So let's try. The moment you see instead of tumor is cancer, that means the end stage. What are always the key phrase that we say for this cancer here? Can you tell me, Chloe? What are key things that we always can relate with cancer? Like some characteristics cancer cells will always have. Hmm? No, I mean like what are some characteristics? You're not answering the question. I'm saying what are some characteristics of cancer cells? How come I know a cancer cells is cancerous? So what are some characteristics? Cancer cells that is not considered tumor. Cancer cells what? What else? The M what? Cancer cells can? That tumor cells cannot undergo. No, cancer cells can metastasize. These are always the key phrase we say, where you actually can break loose and invade your this bloodstream and secondary to form secondary tumors in distant locations of your body. Yes, we also have angiogenesis, where you have development of your this blood vessels towards itself. You always will, however, realize that metastasis is the very common word that we have instead. So if you want to say cancer, you must further elaborate on this. But one thing both cancer and tumor share in common is what phrase? Can you tell me, Megan? They always have? Of? Single? Yeah. I'm not talking about accumulation of mutation within a single cell. I'm talking about accumulation of mutations within a single cell lineage. Understand? That means within your skin layer, with a particular layer of cells undergo multiple mutations. That's why that cells eventually will become a tumor. Alright? Again, when you say accumulation of mutation, similarly, that both always must have is what? You must tell me what mutations you occur. So what are the two major mutations must occur to at least form a tumor, Myra? Mutation of what? Tumor suppressor, or why you're gain a function? Isn't that a good thing if you're looking? Yeah. How many? No, at least one. Okay? Alright, POG, which is your proton oncogene. Please take note, I'm not talking about alleles, I'm talking about genes. That means one single gene, right, is a proton oncogene. Okay? Remember, proton oncogene is not just your RAS protein. What does proton oncogene code for? Any idea, Colentia? Yes, it's any proteins that will actually stimulate cell division. That means the receptor, right, that receives your growth hormone, that brings about cell division, that receptor can be a proton oncogene. Or maybe let's say your this protein kinases that are involved in your this cell signaling to stimulate cell division is also a proton oncogene. Or maybe anything, okay, can be your checkpoints, okay, that it will help you bring about cell progression is also your proton oncogene. There are any proteins that stimulate cell division, not cell growth, cell proliferation. And then what else? What other mutation must I also have, Myra? Loss of function mutation of what? How many? Yes. Several tumor suppressor genes. When I say several tumor suppressor genes, I'm not talking about the alleles. Doesn't mean that, oh, I must have two mutations on the allele. No, tumor suppressor genes are genes. Alleles are alleles. One gene can have many alleles. But I'm not talking about gene. Tumor suppressor gene is like, if you have P53 or CAROS, or all this, you have many different types of genes that encode proteins that will either promote apoptosis or again promote DNA repair or actually arrest your cell cycle. If you have many of your this tumor suppressor genes that perform your this plethora function and prevent tumors, they undergo mutation, this is also going to develop cancer. So I want to see at least one and many. These are the basic things. Will they form cancer yet? No. At most they form tumors. So what's the effect if you actually have all this eventually? It will result in what kind of cell division that we always say? And then what else at this stage? And they have contact inhibition or no contact inhibition? And have no contact inhibition. Who here do not know what's contact inhibition? Okay, contact inhibition is usually cells will divide. But the moment one cell touches another cell, in contact with another cell, it stimulates them to stop dividing. Contact inhibition, that means you stop dividing upon contact with another cell. Understand? But in your cancer cell, tumor cells, usually even though they are side by side, touching each other, but they're still stimulated to undergo cell division. Understand? So this will allow for accumulation of more mutation. Cancer critical genes. Example, telomerase. Okay? But let's see, because we need to relate what kind of mutation can lead to cancer. So since we already studied these various types of mutation like gene and chromosomal mutation, we need to take note what are the genes do we want to focus on to undergo mutation and what's the significance of it. Since we already know how it can form cancer, so let's try. The first gene that we actually can have mutation, you should know, is your POC. So we have multiple POC. Let's see. So proto-oncogenes, let's learn the difference between proto-oncogenes and tumor suppressor genes first. 1, 2, 3. First difference between proto-oncogenes and tumor suppressor genes are what, dear? Proto-oncogenes? Proteins that stimulate cell division, well? Not only inhibit cell division, but also promote and as well as promote enzymes that undergo DNA repair. Okay, so again, the famous tumor suppressor genes you all should know. Under your cancer chapter is on the last page of your notes. P53 is just an example of a tumor suppressor gene. But there are other tumor suppressor genes you need to take note of. How do I recognize? You can see the roles they have is that they either inhibit cell division by promoting apoptosis, or arresting cell division so you prevent the next progression of cell cycle, or you promote enzymes that undergo DNA repair. Another tip you can see is that usually for your tumor suppressor genes, if you undergo mutation, is it act recessively or dominantly? Tumor suppressor gene. That means both of the alleles of the gene. So both alleles of a gene must undergo mutation. Then you can knock out. Whereas proto-oncogene, how do I recognize that the question is testing me on proto-oncogene? You need only one. One of the two undergo mutation, then you will form this cancer or tumor already. Understand? What's another difference between proto-oncogenes and tumor suppressor genes? I want to ask, if you want to have this tumor suppressor genes to develop cancer, is loss of function or gain of function? How many? Both? Multiple. I'm talking about genes and alleles are different things, okay? Whereas proto-oncogenes, so you need to have at least, so you're right, is gain of function of at least one gene. Understand? Okay, come, let's go through. So first one we need to learn is what are the things that can bring about here. So for your POC, let's talk about your structural mutation first. First one you should know is your translocation mutation. This is for your essay. Translocation mutation, you should know is a type of structural mutation here. If I want to describe translocation mutation, is that when a section of your this chromosome breaks free and then attaches to a non-homologous chromosome. I'll just write down for the reasoning because you should know. Definition in essay is always needed. Some of you never even write definition. Okay? Now, very badly done for the essay. You will see why. Shit, what I want to write? When a section of a chromosome breaks off and attaches to a non-homologous chromosome. Idea how is it related to POC? So it's related to POC is because let's say the proto-oncogene may be moved to a different location, alright, where it's now placed under the control of enhancers or an active promoter. Let's try, ready? Enhancers are again DNA sequence or proteins. Chloe? Enhancers are DNA sequences or proteins. Then what are activators? DNA sequences. So I repeat again. Enhancers are DNA sequences or proteins. DNA sequences. Repressors are DNA sequences or proteins. Then what are silencers? So enhancers and silencers are DNA sequences. What binds to enhancers? And what binds to silencers? Repressors. Remember, 1, 2, 3. How does enhancers upregulate transcription? Number 1, recruit. Recruit. DNA, bending, proteins, and then what? To bend the yes, to increase what? In order to increase what? Nani surface area. I mean increase interaction between activators and other transcription factors. I'm asking you to recite to me the keywords. Then what do you also say? Huh? What's the keyword you're missing? Increase stability of what?
Yes.
Transcription initiation complex. Again, from the top. What's the first thing? Record. 2. And then what? 2, increase between activators and other transcription factors. Okay, and then finally, what do you say? This will increase of yes, transcription initiation complex. Okay, duplication you should know already involves replication. So this is replicated, okay, resulting in increased numbers of pop copies. Okay. So when I increase number of pop copies, or you say like the sequences are repeated, what does this actually lead to? Again, imagine you have multiple copies of your this proto-oncogene. The multiple copies of proto-oncogene will lead to increased expression. Or you can say you will have excessive gene production. Okay? So again, multiple copies of pop. Alright, so you increase expression or you excess gene production. Duplication. What else do I also have besides your this translocation and duplication? I also can have aneuploidy, right? So aneuploidy is chromosomal number so what can lead to your this one? So same thing, multiple copies due to non-disjunction. Gene product. So you can see how it's related, all the mutation can be related to cancer. Again, due to non-disjunction, either you increase expression or you produce excess. Another one, we also can have gene mutation. What kind of gene mutation I can have? Two ways. Number one, you can have gene mutation that occur in promoter, enhancer, or silencer. Remember, you're trying to increase expression, huh? Okay? Increase in promoter, enhancer, and silencer that controls the proto-oncogene, controls pop. So that's why, again, you can say how this will cause the increase in expression. Increasing its expression. So this is for, again, mutation in promoter, enhancer, and silencer. I also can have, again, mutation. Okay, in this such that it's hyperactive or resistant to your this degradation. Like for example, maybe you have mutation in my this G protein coupled receptor, such that even without the growth hormone ligand, your this receptor is actually activated. Okay, so that can be one. So you can have mutation, all right, in the coding region, or you can say exons, okay, of POG, altering its 3D. And also, this can mean that it is more hyperactive, or you can say resistant to degradation. Like for example, it may be the G protein can never be actually converted. There are many ways that your cell signaling does not shut down. So we have mutation in the exons, okay, so it's now hyperactive or resistant to degradation. So this is your POC. Right, now besides POC, we also know we can have loss of function mutation. So I also want to include more. So translocation, I'm lazy, I'll just write this and replace this. So you should know, I also have tumor suppressor genes. You can have translocation and tumor suppressor genes. So this time a section it breaks apart. So the idea is, this time you're not actually just breaking off, you are just placing under the location, the control of silencers or a less active promoter. So again, the tumor suppressor gene, okay, it's moved to a different location, but this time you place under the control of silencers, or you can say a less active promoter. Okay, it does not stimulate cell division, but more likely you're trying to inhibit cell division. Alright, it will no longer be duplication. We can have deletion. So in a deletion mutation, what happens is that you have loss of a section of chromosome. Alright, resulting in decreased number of copies of the tumor suppressor gene. So what happens is that you should know loss of your DIS2. So I repeat, so it breaks, and then later this loss of tumor suppressor gene in the delete section, you will have instead of increased expression, you will have again lack of expression. Okay? Or you can say no gene product is synthesized. Alright? Another thing. Can I erase the top? I need to write inversion. Inversion is also another one that is a mutation. So when I have an inversion mutation, so hopefully you know what inversion is when a segment of nucleotide sequence breaks off and then rejoins in a reverse manner. Okay, so again, so what happens is, how come you're not moving to a different location anymore? So when you have such inversion mutation, you should know that your nucleotide sequence is being read in a reverse manner. Okay? This is bad is because that again this amino acid sequence is incorrectly read, and therefore you result in the formation of non-functional. Tumor suppressor gene product. Next one, aneuploidy. Aneuploidy are right here. Non-disjunctional. Idea is you have loss of copies. So have lack of expression of this tumor suppressor genes. Residocytes, okay, that means no gene products at all. Okay, so in other words, you are no longer able to inhibit, oh, I forgot, right? To inhibit, okay, cell division or promote apoptosis. Okay, gene mutation. Gene mutation can have this. So we also can have mutation in your promoter, enhancer, and silencer. They are both acceptable, which controls your tumor suppressor genes, and this decrease its expression. We also can have mutation in exons, but this time you don't tell me it will form hyperactive or resistant. When you form exons, you're making it non-functional. The idea is that you want to have loss of function mutation. Okay? Okay? So you know cancer, when you have all this deletion and insertion mutation, they can actually cause either gain of function mutation or loss of function mutation. You can actually take note why mutation is a chapter that is usually linked to cancer. Cancer, however, has some similarities to another healthy adult cell. What is that, Wani?
Gamete cell.
Which and
Stem cell.
stem cells. Ready? Now, gamete and stem cell, there are some things in common, right? Sorry, I mean, cancer cell and stem cells have something in common, right? 1, 2, 3, 4. What is the first similarity they might share in common?
Telomerase gene is activated.
Telomerase gene is activated. Okay, so what does telomerase do?
It
Elongates.
elongates the telomere.
Question, Kalantia, what's the standard phrase that we describe for the role of telomeres? Telomeres will act as protective. You know they always got N replication problem. So how do you protect your genes? So no, I want to hear the role of your telomeres. The standard answer you tend to write for exam. They act as protective buffers to prevent loss of genes due to N replication problem. Again, get ready, you tell me the second one. Tell me what's the number one factor for telomeres? Just now you just repeat. Act as
Protective buffers.
to prevent. I'm listening.
To prevent N replication problems.
Your head loss of genes due to N replication problem. So how do you write for exam? Is what they act as
Protective
to
buffers.
prevent
Loss of genes.
due to.
N replication problems.
Yes, you. Can you now tell me what's the next thing we need to take note for the function?
Protect from degradation by enzyme cells.
How? Because they associate with what kind of proteins? Associate what kind of proteins? Telomeric proteins to loop the DNA. Why? And that will help to protect against degradation by? What's the second reason? What does that do? Also prevent N joining off, because you know there's always an overhang. We don't want the chromosomes that are non-homologous to adhere to each other. That will affect gamete formation. Last one. When shortened,
When shortened, it will stimulate apoptosis as it reaches the Hayflick.
so when shortened to Hayflick limit or the minimum critical length, it will trigger the cell to undergo apoptosis, because it's trying to tell the cell, hey, the moment you undergo the next round of cell division, you'll start to lose my essential genes. Enough time to think. What's another similarity between your these stem cells and cancer cells? They both undergo what kind of cell division? Sorry? What kind of cell division? You know there's only two types, right? What's the two types of cell division? Both M and? So both undergo? Yes. Next one, Megan, what's another similarity? Both are? Both stem cell and cancer cells are undifferentiated. Next one, what's another similarity, Myra? Both are able to, because they're active telomerase, they can undergo cell division for long or short? Because they have active telomerase, right? Would they ever shorten the Hayflick limit? No. So again, they are able to divide for a long period of time, but they are not the same, right? Let's see what the differences between stem cells and why stem cells will not become cancer cells. Can you tell me, Kalantia, what's the first difference between stem cell and cancer cells? Stem cells, when they divide, do they have dysregulation and checkpoints? You know what checkpoints? So, do they have dysregulation? So stem cells can become cancer.
They have checkpoints.
Sorry?
They have checkpoints.
So what kind of cell division would they have? Uncontrollable? Controlled cell division. Whereas cancer cells? Uncontrollable cell division. What's another difference?
Stem cells got contact inhibition.
While
Cancer cells don't have.
no contact inhibition. What's another one? Stem cells, they can, because they have active tumor suppressor genes, right?
Yeah.
So they can undergo.
Apoptosis.
Whereas,
cancer cells are not able to undergo apoptosis.
yes, because they have loss of function of many tumor suppressor genes. Next one, Chloe, what's another thing? For cancer cells, are they able to differentiate? Then how you form more white blood cell? Your bone marrow stem cells cannot differentiate. So, stem cells can what? Okay, to become specialized cells. How about cancer cells? They remain undifferentiated. Understand? Next one. Megan, what else do we know that cancer cells undergo but stem cells cannot? Stem cells, they do not undergo. Where's cancer cells? What else, Myra? Cancer cells, they can undergo. And which means that they can actually grow blood vessels towards themselves, whereas the stem cells cannot undergo angiogenesis. Okay? Next one, can you tell me what's another difference? You know what is anchorage dependent and independent? So which one is anchorage independent, which one is anchorage dependent?
Stem cells is anchorage dependent.
Whereas
Cancer cells is
anchorage independent. Very good. The idea of anchorage dependent means you usually anchor towards your own tissue layer. They will not break loose. Okay, they anchor. But you know cancer cells, they can break loose. So it means that they are anchorage independent. Understand? What else? We know that this normal stem cells, okay, they again have the same chromosome number each time they divide. But cancer cells may have chromosomal aberration. What? There may be aneuploidy. There may be structural mutation. Agree? Things like that. Last one, checkpoints, well regulated. The other one, checkpoints, dysregulated. Learn all this. Okay? So that's why they are not the same. Okay. Now. Let's press down. Okay. Now I want to do respective questions. Alright, each of you will do your respective questions for me to mark. But before you actually help me do, can you help me start these questions in the worksheets before I forget? Number 1, can you help me start 1A? Okay. Star 1A, star 2A, 2B, 2C, 2D. Entire of question 2. Please also help me star question 3C part 1 as well as 4A and 4B. Okay, now each one of you will do your respective questions for me to mark. Those you, Wani, Megan and Kalantia, I want you to do question 1A and 1C. The rest of you, do you see question 3A and 3B? I want you to do 3A and 3B because these are all about your data analysis. Please use the other questions, the answers as a revision material. I'll send you other answers as well. Try this, then later I can do molecular techniques. Once you're done, let me mark. Here, who haven't done the RI paper 3 2023? Have you all done the prelim paper RI paper 2023? Prelim. Oh, so I want to give another round on paper 3. But if you haven't completed, if you completed, I won't be printing out. But those who already completed, you can use me to mark. So it looks like this. Okay, let me confirm. Yep, it looks like this. Have you all done this paper 3? No? None of you done? Okay, I'll be printing out. You all can actually try. Want me to mark and critique your way of writing? You can use this as a time of revision now. Girls who did this practical paper, please, paper 3, okay, practice paper, okay, you all know there's this one called the immunity. Please go and study the answers. The answers, right, the key thing they bold and underline is usually the phrases how you actually have vaccination that will develop your this antibodies. Do not straight away tell me about how you form your this plasma B cells. You need to tell me how you even form APC first. I want to see keywords like your MHC peptide complexes, which you are lacking, all of you are lacking down there, and how you have your this activation and differentiation to form T helper cells, and eventually activation and differentiation to form your plasma B cells. All this you are missing the keywords here. So go and study the answers. The rest of you go and also take note. There is actually a question, and the answer key is actually the key thing that you need to focus on for that practice paper. Okay? done? Let me mark. Whoever is done, I can mark. Let's see. Again, very good. 3 yourself. Now, do you know what evaluate means? Do you need to give a stand if you want to give your evaluation? Yes, you need to give a stand, and the evidence you always have to say is going to support your stand now. So you say you have lower rate of survival. Question is, do you need to quote data? I said before, evaluate, explain. As long as with reference to the data, you definitely need to quote data. So you number one, you didn't actually quote the data, and you never tell me are you supporting or not supporting. This is support. You support. So you support, and then you will say that how decrease with high gene have lower survival rate than that of low gene every year. Now, here, right, again, I can accept if you just say support. Please say your stand that you support, but you never quote data. So I cannot give you maximum marks, understand? What is this? Question is, has been identified what's an on-call gene. Now, on-call gene is again formed from what? So that's the missing one that you're missing from. So again, proto-on-call gene will always mutate to undergo what kind of mutation to become an on-call gene? Gain of function. That is the missing point. And then you will say how you will either have not just stimulate cell division, because that will mean it's a proto-on-call gene. What? Wrong? No, not uncontrolled. Excessive overexpression, things like that. But the keyword in the question is you need to take note what kind of cell division will it lead to. Uncontrollable cell division, and then that's why it can develop in cancer. So I cannot give you a single mark here. Okay? Who else is done? Those who are waiting, you can try the other question now. So, what you were trying to say? Across what? This is the quotation of data. Fine, you want to say hypothesis, correct? So will it actually lead to cancer? Yes. So overexpression, increased chances of death. Wait, usually survival rate. Sure, fine. How do I have what kind of mutation? Sorry, what kind of mutation? You are missing the keyword. Where? Gain of function mutation. You didn't write that. So you never write gain of function mutation from. So proto-on-call gene gain of function mutation to become on-call gene. That's the keyword. And you also must tell me from this one. So again, this leads to, again, you say lead to uncontrollable cell division. So why would I have uncontrollable cell division? Because when you have this, you have high, you never relate. It led to overexpression, so you will have uncontrollable cell division and it can become cancer. Because oncogene is trying to say that it's becoming cancerous. Understand? Always look at the keyword. Who else is done? Okay, please complete. If you want me to mark, complete by your next class so I can actually mark for you. First here, the survivor. You telling me that this shows, again, quotation and data, fine, I can give you 1 mark, but it doesn't show comparison. Idea you should be actually, when you want to tell me data, you tell me based on trends. Okay? So as the number of years increase, so the idea is every single timeline and every single timeline in the survival rate is all decreasing. Okay, survival rate. Okay, so fine. Mutation of data, you compare with high, sure, low. You must support, take a stand. So you want to say support, I'll give you the stand. Gene that sure, gain or function mutation of a protein oncogene. Excessive cell division. How come you have excessive cell division? You never relate. The one that you are supposed to relate is because they show you gene expression. So you must tell me this causes excessive expression of the gene that led to uncontrollable cell division, which is the missing point that you have. That's why I cannot give you the full mark. Who else is done? By the way, for those who did question 3, the question was question 1A and 1C. So make sure you know how to do question 1A and 1C, and then later I'll go through. Then after that, I need to do more tech. Sorry. Not bad. That just now I asked you the 4 reasons when you have gene mutation. The gene mutation can lead to side, and now the missense also, and then also if you have mutation in your disintrons, also a gene mutation, right? But what happens to introns after that? Think about that. Those who don't have Mogen, take one. Mogen, molecular technique. So you say cellular mutation. You're talking about protein production or protein structure? Or conformation? Conformation, dear. Do you know why I focus on conformation and not production? Think about this, they code for amino acid sequence, which will affect your amino acid of your polypeptide chain, which will affect your conformation here. You talk about production can be level of expression. Level of expression may change. Do you know that? Maybe you put it in near active promoter. Then level of expression change. That's called protein production. You understand? So for me, right, is that your issue right now, you need to learn how to use the right terminologies. So for you, if you go and see your practice paper, sometimes right, your phrasing is really cannot be accepted. It's because you lack the keywords that you have in your answering. So again, introns are its size. Not primary, do you know what primary mRNA is? Mature mRNA. Pre-mRNA is the primary mRNA. We call it mature mRNA. Understand? So again, protein product still retain their original function. Fine. Say conformation. Always focus on conformation first. Why protein product remain function? So again, conformation there. Always write conformation. I don't care about the rest.
But how do you relate it back to cancer?
No, you're just saying that how come it will not lead to cancer? You just say the protein function remains intact. Idea is that all this, that the protein function is not mutated, or the protein conformation is not mutated. So function is still the same. They keep their function. So triple suppressor gene will not lose the function. The proton oncogene will not become a proton oncogene one. Understand? Sorry, will not become an oncogene one. So again, you need to talk about this. Then I can give you the credit. Yeah. Percentage increases from. Percentage increases. So it decreases. Decreases from here to here, and then percentage increase. Oh, sure. Good. Explain. But wasn't it the in metaphase is arrested at metaphase?
Oh, really?
Because you cannot undergo metaphase. You see, the ratio of number of cells in anaphase decreases. Yeah, so there are lesser cells undergoing anaphase. Okay, there are more cells undergoing metaphase. Metaphase is a checkpoint one. So we kind of show, we kind of try to show you that they are trying to being arrested at your metaphase.
Oh.
You understand? So, no.
Okay.
Who else?
Can I go for
Oh, you sure? Megan? Done? No? Valentia, hopefully you know, look at the ratio of your these cells undergoing metaphase and anaphase. The ratio of anaphase is to metaphase is decreasing, right? More cells undergoing anaphase or metaphase? Metaphase. The question is, how come this shows more cells are undergoing metaphase? Shouldn't it be almost the same? Means they are most likely arrested at metaphase. And metaphase, what checkpoint do I actually have there? Do you recall? M checkpoint. So during M checkpoint, do you think the M checkpoint is able to bypass? So most likely, you can see, So that's why you can see why centromeres doesn't seem to be dividing. When centromeres do not divide, again, your spindle fibers cannot shorten. That's why you actually cannot separate your these chromosomes or chromatids, to opposite poles of the cell. Okay, things like that. Rest of you are done, please go and ponder. You'll be helping me with 1A for those who never do question 1. Very good. Hey, you only gave me 3 reasons left. So keyword is 4 reasons left. Why you only give me 3? So what's the last reason? Non-essential amino acid. No, the question is how come they are arrested at M checkpoint? Or what happens? Now, you didn't mention to me how come they'll be arrested at M checkpoint, because the question is asking you explain. So, if they are actually arrested, it means why they can prevent cancer. Because this centromeres will not, or your spindle fibers cannot shorten, or you cannot actually pull your sister chromatids to opposite poles of the cell. Understand? My mark there. Okay. Use the word excise. You are splicing axons. You use the wrong terminology, I cannot give you credit. Okay? So they will not splice up, then what? What do you mean? If you splice up, learn how to elaborate. So why you can form a functional protein? Because the mRNA or the amino acid sequence remains the same. So when your amino acid remains the same, conformation of protein? Yeah, so that's why function doesn't change. I always want you to elaborate step by step. Okay, so this is the missing one. Gene mutation occurs in non-critical region. What do you mean does not affect protein form? It does. It can affect it. Maybe you change outside. But again, you say non-critical region, that means what? The amino acid must be what? So you never relate to the amino acid. So idea is that the amino acid will be a non-essential amino acid. That's the keyword that is related. Missing that keyword, even though you have the same meaning, I will not give you the credit. Again, then once you say this, what happens to the function? Function of protein remains unchanged. It will still affect the protein form, but it's just that function remains intact. Genetic code is degenerate. So what kind of mutation? Silent mutation. You have to write the word. The mutation may be a silent mutation. You understand? Same protein form. You must write silent now. So a lot of things is that you are missing the key thing. And purposely I'm not giving you credit, because if I give you credit, that means I condone this. Okay? Another one, you say same properties. Wrong. I will not accept same properties. I need the keyword R group properties. Okay? So that's why you form similar R group interaction. Because you write same properties is wrong. Not exactly all the same. You also know iodine and then chlorine, different size. Even though they can form ID, the properties are slightly different. So not exactly same properties. So again, almost the same protein structure. If you have this, then I can give you a credit. So for you, it's all either wrong terminology or missing key phrase. You understand? Describe concentration, percentage increase, ratio, decrease, right? There are more cells undergoing mitosis. Not really what? But the mitosis are being arrested at anaphase. Cannot bypass, the same thing. Reason is if they are unable to align properly, so how come it will not lead to cancer? So you need to take note how come it will actually decrease the amount of cancer cells, because your sister chromatids can no longer be separated. Okay, so this is the missing thing. Now, can you help me pass down answer sheet? Thank you. Now let's go through the questions here. The first one I ask you all to do is 1A. Again, this is an explain question. I said before, any explain question, we either use the specific keyword technique or the overview point technique. So my question is, for this particular question, can you tell me, Chloe, do I use specific keywords or overview point? We know the specific keywords, obviously 4 marks is 4 reasons, but what is the keyword we are looking out for? Why is never a keyword. Why is like explain. Explain, describe, and comment, or discuss are all command words. Okay? They are not keywords. Keywords are those that let you know how the marks are delegated accordingly. So what do you think a keywords is? Try again. Gene mutation. So you need to tell me gene mutation, okay? Then obviously link to cancer formation. But as long as you tell me the 4 mutations that can occur in the genes, yet you still can form functional protein, I will give you the credit here. Okay? So, idea is, this is gene mutation in your this may not always lead to cancer. Okay, can. So one thing I will accept is that this actually is cancer formation is the next keyword, right? So you need to relate to cancer formation, if not it will be marked down. So first reason why your gene mutation may not always lead to cancer, let's ask Chloe, why if I have mutation in my genes yet I will not form cancerous cells? What's the keyword related? So silent mutation, very good. Gene mutation can lead to silent mutation is the first key phrase, and then due to? Where? Different codon? Due to? Effect. Whole thing. I want to see this entire thing. If it's not in your notes, answer sheet, please help me include. So I want to see a silent mutation. I'm very strict, by the way, because I want to make sure there's no room that teachers can actually mark you down. Alright, so you can say how is silent mutation where the same codon can code for a different codon can code for the same amino acid. Alright, since the genetic code is degenerate. So I want to see degenerate. I want to see different codon can still code for the same amino acid. I want to see the keyword silent mutation. Without this 3K phrase, I will not give you 1 mark. Okay, second reason is what, Myra? Since I never get to mark for you. Let's say change codon code for a missense mutation. But what about the amino acid that is substituted?
Can be substituted
With similar
with similar
properties. Highlight the keyword similar R group properties. They're missing the keyword called missense mutation, but it's a different amino acid with similar R group properties. Don't write properties, there are different kind of properties. Okay, however, you must tell me, I don't want to accept just affecting protein structure. You must say either you do not affect the conformation of protein structure, you must say how the protein remains functional. I still need to see the word protein remains functional. Another mutation, Vivian, can you tell me what can I also accept? The amino acid substituted is a non-essential. So the idea is we can actually substitute a non-essential amino acid. So same thing, I want to see the keyword. No change in 3D conformation and protein structure and function. So I want to see structure and function all together. Last one I also can accept is you say mutation in your introns. Introns are part of genes. It's on your DNA. But you can say how introns, please don't say splice out, please use the word excise. And it's not primary RNA. Primary RNA is called pre-mRNA. So you excise from the pre-mRNA to form the mature mRNA. So you can say how, keyword they are missing from the answer sheet that I feel they should have actually written that down. Is that, again, you must say how the amino acid sequence in polypeptide chain is unchanged. Protein structure and function unchanged. Always relate with this. Okay? Clear? Okay, anyway, moving on. I can accept the others, okay, up to you. But again, let me look at 2C. 2C, please take note. Do you see with reference to? The moment we are with reference to, can you tell me, Chloe, what must I always do? Code relevant data. I want exact data. Notice how there's 2 marks. What is the 2 relevant data? The effect of this drug on, again, the percentage of cells, and again, the effect of this drug concentration on the ratio and number of cells in anaphase to your metaphase. Okay, so again, as long as you code data, I think the answer sheet is quite self-explanatory. I will give you 1 mark for each relationship between the concentration of percentage of cells in mitosis and the concentration of percentage ratio of cells in anaphase and metaphase. Okay, now what I want to talk about is part 2. They ask you to explain how come if I have increasing concentration of this, your this metaphase and anaphase and will actually the ratio will actually decrease. Alright, now again, you should know already if you have decreasing ratio of anaphase and metaphase. By right, it means that there are more cells undergoing metaphase or more cells undergoing anaphase. Myra? Look at the ratio. Ratio of anaphase is to metaphase. It's decreasing, right? More cells undergoing metaphase or more cells undergoing anaphase? Metaphase. If more cells undergoing metaphase but yet they cannot undergo anaphase, means what is arrested? They are arrested at checkpoint. So you need to explain. As long as you tell me they are arrested at M checkpoint, okay, that's why again this actually cannot go past my metaphase to anaphase. I'll give you 1 mark. However, you need to elaborate because explain means what? Either use the specific keyword technique or the overview point technique. Isn't this using the overview point technique? The moment you tell me they're arrested at M checkpoint, then what does it mean? It means that either you tell me centromeres are not separated, or you tell me spindle fibers cannot shorten, or you can tell me this chromatids cannot separate to opposite poles of a cell for anaphase to occur. You are already answering the question. So I repeat, when I say overview point, the first point is always answer directly to the question, and then subsequent is just elaboration on that. Okay? Anyway, can we go through this question 3? Question 3a, they asked me calculate the percentage increase. You all should know it's just mainly the difference. I'm not going to go through because answer sheet is quite self-explanatory. What I want to go through is your B part 1 and 2. From now onwards, you see evaluate. You see the command word. Comment means you have to take a stand. Megan, because I never marked for you, which is the command word we see that we have to give both sides of the coin? What is the command word in contrast? That I know I have to give both sides of the coin. Because I know comment and evaluate, I need to take a stand. Whether I support or I don't support, I agree or I disagree. So which one do we not take a stand but give both sides of the coin? Not sure? You? Discuss. If you see the command word called discuss, means you have to give me both sides of the coin. You tell me this one yes, 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 and the other one tell me no, no, no. So Megan, your essay, you tell me agree and disagree, I will not give you the credit for your disagree. I only choose one. Okay? So, again, in this case, they ask you evaluate the evidence using the data. You know 1 mark is about quotation of data. I say so many times already. But there is never right or wrong answer for evaluate and comment. They will always have both answers that are acceptable. Usually the famous answer, I would say this do not support. Why? Because even though you see the data we have there, relation doesn't lead to causation. So please take note, correlation does not mean causation. You all learn your H2 math, right? This correlation and regression. Sometimes you have your R may be less than 1. So it doesn't mean you have correlation leads to causation. Can you highlight that key phrase? That is usually the very common phrase if you want to disagree. It doesn't mean that, oh, right now I did this data. Okay, I see that if I have increasing, expression of my CDKN3, the survival rate actually will decrease. Things like this. Because there may be other factors at play here that we never investigate. We are just investigating this one. But like I said, for comment, usually both sides of coin I will accept. So if you actually say agree, as long as you tell me that you have again overexpression results in decreased rate, across all period times, and you quote data, I will give you the full 2 marks. Understand? I'm just trying to show you how we tackle this data analysis. Next one, keyword in the question is called oncogene. You should know oncogene. Key phrase we always describe is it must be a gain of function mutation from a proto-oncogene. Without the word gain of function, highlight in the answer sheet. Without the word from the proto-oncogene, I cannot give you the first mark. That's an idea for oncogene. However, please don't tell me, oh, it will stimulate cell division. No, because oncogene means you want to become cancerous already. Show me how it becomes cancerous. Elaborate with effort here. So you must tell me how you will have overproduction of your gene products. Leading to keyword. How will it lead? Uncontrollable cell division. Because your oncogene, you have over amounts of cell division. And hence cancer. Clear? Without the word uncontrollable cell division, no marks. Alright, clear? The rest of it, you can use this as studying material. Answers is already given to you. I want to do this PCR and your gel electrophoresis and your solvent blocking. So for molecular techniques, we are always focused on this 3. I'll only focus on the main things for this chapter. Again, for those who never do well for the PCR question in the practice paper, please learn the steps. Now, one famous thing about PCR, I'll go through a bit about PCR techniques. But you should know the PCR, they always like to compare with DNA replication. Okay, now in your notes, you can actually write that down. But let's ask yourself first. In PCR, we learned there is always 3 temperatures. Let's go through. I say already, the temperatures just follow your school. Different school got different values. But can I ask Chloe, what is the temperature first? Always start out with what first? Sorry. Yes. If you write 92 degrees, I also accept. Do you know what occurs during the 95 degrees Celsius there? What occurs during 95 degrees Celsius is denaturation. Do you know what denaturation is all about there? Your head ah? No lah. It's what? Your double-stranded DNA separate to become? By breaking what? So you saw, if you never write by breaking hydrogen bonding between complementary base pairs, I cannot give you the credit. Chloe is on the second page of your notes if you don't know the steps. Okay, so you all see the second page. Please know denaturation, you will separate the double-stranded DNA. Then the next temperature is what, Myra? Now, 55 degrees Celsius. Now, I'm very astounded when I was marking the practice paper. At 55 degrees Celsius, and then later, right, they start at 55. The 55 degrees Celsius, right, is not about what do you call it? Oh wait, I want to point out here. At the 95 degrees Celsius, some of you tell me Taq polymerase or denature. How can Taq polymerase denature? Taq polymerase is heat resistant so it's resistant to very high temperatures so it doesn't denature here. When I say denaturation, it's denaturation of double-stranded DNA. 55 degrees Celsius, why must lower? Because only at lower temperature, because just now you show it separate, right? Only at lower temperature, you can anneal your DNA primers. Why I must anneal my DNA primers? Because your hydrogen bonds can only be formed then. If you're too high, then you cannot form the hydrogen bonds. What? You understand? And then next one is what? Your 72 degrees Celsius. 72 degrees Celsius is when you have elongation by your Taq polymerase. Your DNA elongation, this is near, I never say it's optimal, it's near optimum temperature of your Taq polymerase, okay, for your this elongation process. It doesn't denature at 95, please, it's thermal resistant, alright? Now, speaking of this, I want to realize, because I realize some of you don't even know how to draw the reverse and the whatever primer. You know your this PCR involves 2 primers, right? Reverse and forward. I'm going to give arbitrary. So this is on a single template DNA strand. Let's see. You all should know what are reverse primers and what are your this forward primers. We have 2 different primers here. Is to flank the DNA. So the forward primer and the reverse primer, one thing that I fail to see in some students is you all never tell me direction. You always must showcase direction for me. Without showcasing direction, you will be penalized. A second thing is, again, you should know this is the location where your this forward primer will first bind to on this first template DNA. Agree? Do you think the reverse primer will bind to the same DNA strand? No, what? The other strand of DNA must be complementary, right? So if you were to see, the other strand of DNA will be your TAAG, TACG, and then TGG, CTAA, T. You all fail to tell me, for some of you, that there are 2 strands because the question like to show only 1 DNA strand. But you should know under denaturation, there is always double strand that just separated. This is where the forward primer will bind to. On the other strand is where the reverse primer will actually bind to. So now let's see. If forward primer must bind to here, what is it going to be, Chloe? What will be the forward primer that binds to that? What's the nucleotide sequence? TA, A. Some students tell me the complementary sequence of this. I shook it. Hello, please Alright. So this one will be what? The reverse primer. Reverse primer, you all must draw this one. And then you draw the alternating. That's why here is going to be the 3' end, right? Where it binds, right? For here. That's why this here is A. A G, C, C, G, A. T, T, A. Isn't it exactly the same as this? I want to see direction. Please know how to draw forward and reverse primer. Okay, anyway, I need to quickly move on. So this one, we need to take note about two things. First one is the difference between PCR and your DNA replication. Hopefully you know your PCR, they will involve your tag polymerase. This one is DNA polymerase. Some schools use DNA polymerase 3 and some schools use DNA polymerase 1. Megan, your school is one of them. DNA polymerase 3, what does it do? Oh wait. Catalyze. Phospho. To elongate the entire DNA, right? Another one, also a proofreading? Have. And then DNA polymerase 1, what is the role? What's the role for DNA polymerase 1? Hydrolyze RNA primer. Your school went through this what? So please know okay? So this one, however, is not heat resistant. Have proofreading due to presence of 3' to 5' exonuclease and 5' to 3' exonuclease. Yes, you know. This one must be 2 strand forward bind here, backward bind here. But the question they always like to show only 1 strand. And students think that all the forward and backward primer bind to the same strand. Then they're careless with this. Okay? So this one is something we need to come up with. Now the difference is, again, the primers. Please take note, I'm using 2 and they are always different. You see the DNA primers here are largely different from each other. Okay? And they are DNA. Your forward and your reverse. Whereas here, we make use of RNA primers. Alright, another difference, we need to take note, DNA. Okay, this one has 3 temperatures. Which you already clearly know, the 3 temperatures down there. This one only 1 temperature. Another difference I can say how, for PCR, your DNA replication is strictly continuous. There is nothing discontinuous. Why? Because the 2 strands are completely separated. Whereas here, your DNA replication is continuous for which kind of strand, Wani?
Leading strand.
And then?
Lagging strand is discontinuous.
Yes. How do I recognize a leading strand from a lagging strand?
Leading is away from origin, then lagging is towards origin of replication.
No.
The other way.
Replication fork. Talk about the replication fork. Leading is again towards the replication fork, and then discontinuous for the lagging, which is away from the replication fork where your helicase is trying to open it up. Can I erase this?
Yeah.
Chloe? Yeah. Now another difference I also can talk about is how you separate your 2 strands of DNA. How do I separate, Chloe? Oh wait, by what? By what? Denaturation is a process. That means you separate your double-stranded DNA is by heating. Understand? So that's why you break hydrogen bonds. But this one separation is via what there? Enzyme start H. Yes, correct. By helicase. Next one. Another difference that we also can talk about is where does it actually where does it actually take place? This is within This is again within the cell. Okay, now when you're ready, I want to talk about this principles of gyro electrophoresis, which is flip, where you see this page of your notes called gyro electrophoresis. Okay, for gyro electrophoresis, I want you to focus on, okay, the three things, which is the principles of gyro electrophoresis, not there. From now onwards, if you ever see a question say what are the principles of gyro electrophoresis, you always write the first one. Megan, is what? Separates DNA fragments based on charge, difference in charge mass ratio. Okay, so I repeat. First one, you always say how gyro electrophoresis separates DNA fragments based on differences on charge mass ratio within a given time. Second key phrase is what, Megan? What kind of gel I use? Agarose gel acts as a... Yes, second key phrase, you're right. Agarose gel acts as a keyword, molecular sieve. Underline molecular sieve for the DNA fragments. So I repeat, agarose gel acts as a molecular sieve for the DNA fragments. And then the third bullet point you will take note is that how does it actually migrate, Megan? DNA is what kind of charge? Net. And then because of phosphate groups, right? And then you always approach which terminal? Positive or negative? Positive. So you say the negatively charged DNA moved towards the positive electrode. Moves towards the positive electrode when an electric current is applied for a fixed duration. When an electric current is applied for a fixed duration. Underline negatively charged DNA, underline positive electrode, underline electric current. Okay, second thing I want to talk about is the ingredients needed for gyro electrophoresis. First needed that I need for gyro electrophoresis, obviously you need the agarose gel. Myra, what did I say what agarose does? Acts as a molecular sieve. So this agarose gel will always be bathed in what we call a buffer solution. Any idea what does the buffer solution or loading buffer contain? Can you tell me, Colantia? What does the loading buffer contain? Because it is under electricity, you need to supply something to act as electrical charges to conduct electricity, which is what? Provide ions. So again, take note. So they actually, you should know, it provides ions, alright, to conduct electricity. Next thing is, we have this thing called this tracking dye as well as your staining dye. An example of a tracking dye we must know is what? Vivian? What is an example of tracking dye in our syllabus to know? Because it's very different from the word staining dye. Phenol. Thymol is an indicator. So you are expected to know at least one, which is the famous one, bromo, phenol, blue. Does it stain the DNA? Your head. It does not. It's not called staining dye already. Now, what is the purpose of tracking dye? Because DNA is actually transparent. When you turn on the electric current, you don't know what's the speed your DNA fragments is going to travel towards the positive electrode. This here travels slightly faster than your DNA. So we know that when I turn on and then this one already approaches the end, you turn off the electricity. So at least I know the DNA fragments has yet to run off. Staining dye. We need to know an example. What's the famous example? Bromide. Ethidium bromide has a one unique feature. What is the phrase how ethidium bromide stains the colorless DNA? keyword that you see under your notes. Do you see the box here that I bold and underline? The word is called intercalates. Intercalates, do you see between the step bases? You just again slot it in the idea. So it intercalates between stacked bases and will fluorescent what color?
Orange red.
In the presence of a trans-illuminator. Understand? So the keyword is intercalates. That's why you can visualize your DNA. So I repeat, role for this you can write is that you actually visualize loading and migration, okay, of your this DNA fragments. We know that this is actually intercalates, okay, between the stack bases of DNA. So that's why it will stain the colorless DNA, okay. Now, next one, what do we also have? We also have my glycerol. Glycerol is another one, part of your loading buffer. Glycerol has one property, is that it's denser than DNA. So the idea is it adds density to allow DNA to sink into the well. So you're trying to make your DNA fragments sink within the well here. So it adds density to sink your DNA into the well. Clear? So please know. Now, why do we study gel electrophoresis? Why do we study your PCR? Because they are part and parcel of this process called Southern blotting. Southern blotting is one important step that you must know very well. It's located on this page of your notes, where you see on the third and fourth column. Okay? So Southern blotting, I want you to take note. Okay? Every step later, you will see, for those who just received a note, is empty space. Okay? Chloe, do you know where? Looks like this. Can you help me flip for her, Wani? Okay, so again, for your this one, why do we perform PCR? Because usually we only have one copy. We try to make many different copies. Okay, so then later you will notice how your first step that you see in step number 1 is that I undergo what we call your this cleavage or cutting of your DNA into fragments by restriction enzymes. Now, after which you notice how, what do I have there? Do you see how to the DNA fragments I will separate by gel electrophoresis, separated by what kind of ratio? Chloe? Charge, mass ratio. Okay, the shorter DNA will actually travel furthest, the longer DNA will travel the shortest distance along the gel electrophoresis. Now, after that, to the gel electrophoresis, what do you see I add, Megan? I add sodium below sodium hydroxide, right? What's the significance? Oh, to those who just received a notes, you'll be writing the significance at the side where you see the steps. What's the purpose of adding sodium hydroxide? Between bases to denature double-stranded DNA. So again, you will write at the side is to NaOH is to break hydrogen bonds between complementary bases to denature this double-stranded DNA. Now after that, why do I need denature? Because later you need to have what purpose? Complementary base pairing with a single-stranded probe, right? So if you didn't denature, your probe cannot attach, okay, to identify the gene of interest. Make sense? Now, the next one is, you notice how before we add the probe, okay, we undergo this addition of the nitrocellulose filter paper over my gel electrophoresis and I apply even pressure. How or what is that process in which fragments can be transferred to the nitrocellulose membrane called, Myra? Capillary? Okay, so at the capillary action, do you see at the purpose down there, that particular step? Which is actually step 4. Okay, so the idea is you can write capillary action pulls DNA fragments out of the gel into the nitrocellulose membrane. Pulls DNA fragments out of the gel onto the nitrocellulose membrane. Underline capillary action. Then after that, you need to bake it to permanently cross-link so it does not fall off your nitrocellulose membrane. And to that, Calentia, do you see we have here this step 5? I now add my radioactive probe, right? Agree? My question is, your radioactive probe usually is how long? And why? How many nucleotides long? That's what I'm asking. 2, 3, value? I'll make a sound prediction. How long? Short or long? Why not ask a simple question? Short or long? Cannot be short. It must be long. So usually the probe, right, is at least 20 nucleotides long of sufficient length. Why cannot be short? You think about this, you want to find your gene of interest. Let's say maybe a BRCA gene that can cause breast cancer. If you use AT, 2 nucleotides long, do you think can bind to everything? The chances of binding to so many nucleotides with the same AT combination is very high, right? But if I use 20 nucleotides long, isn't that going to ensure specificity? Agree? So again, probe must be long enough to ensure specificity. The chances of having 20 nucleotides that is actually the same is very, very low. So again, what's the purpose of a probe? It's complementary base. It will complementary base pairs with the gene of interest. Okay, by annealing to it. Next one. Notice how I need to wash off the membrane to remove your unbound probes. What's the need to do for the washing part? What's the purpose of washing step?
So that like the unbound stuff doesn't show up on
The X-ray film. Yes. So purpose we need washing. Washing step minimize unbound probes being detected on the X-ray film. Minimize unbound probes being detected on the X-ray film. So usually when you want to detect, can I just say, oh, you detect your DNA using autoradiography only? Money? No. Cannot, right? Do you say you only detect your DNA using X-ray film only? No, must always write both of them together. So I want to say I detect your DNA using autoradiography and X-ray film. What is the purpose of using both together, Wani? So what will happen if you have radioactivity? You will cause this X-ray film to turn? Black. Yes. So radioactivity, you can write, turns X-ray film black, which allows revelation of DNA band where probe is bound to. Which allows revelation of DNA band, okay, which probe is bound to. So this is why we perform Southern blotting. I also accept if you use fluorescent probe instead of radioactivity, but you always say fluorescent probe and what? Like for example, we use autoradiography and X-ray film. Can you tell me, Megan, for this fluorescent probe, what will you use? Fluorescent probe and UV light. So the idea is that if you choose to use fluorescent probe, you must say detect under UV light. If you want to say using your radioactivity probes, then you say autoradiography and X-ray film to detect the radioactivity. Understand? Clear? Okay, same thing. We only do selective questions, but because of time, I'm going to ask you to help me star these respective questions first. Then you can use this as a studying material. Ready? Can you help me start? Question 1, part 1, part 3. 1, part 1 and 1, part 3. Also help me start. Question 4. Entire question 4 will help you revise for this chapter actually. But anyway, each one of you, I want you to help me. Do you see we have question 2A? Because of time, we just divide and conquer. Wani and Chloe, you'll be helping me with question 2A and 2B. Then, do you see we have question 3A? Megan and Myra, you will help me with question 3A part 1 and 3A part 2 respectively. Each one of you, do you see 3B part 1 and 3B part 2? So I want to help me with 3B part 1 and 3B part 2 respectively. Please do your respective questions first, so at least I'll give you 5 minutes, then we can complete. go through in 5 minutes time.
Tak tu.
Okay, because of time, I have to go through. Sorry, girls. Can we start off with question 2A? By the way, answer sheet are directly right behind your worksheet now. Already. So we have this investigation for question 2, okay? Trying to study GV. So they use enzymes to cut your DNA molecules into short DNA fragments. By the way, what are these enzymes called, Chloe? Restriction enzymes. So only restriction enzymes can cut, so you can form the sticky ends that you see there, which is also the overhang, right? So now they say, suggest how two structure features of the enzyme used to allow it to cut and perform its function. Structure features is known, don't just tell me site or binding. You need to specify. Is it called active site or is it called binding site? So let's hear. Wani, what is your first structure feature that you have there?
I said the enzyme's active site is complementary, conform in charge to the recognition sequence.
To allow for
To allow for annealing.
binding or annealing. So I can give you 1 mark here. So you tell me the structure feature and you tell me how it's related function. She say active site, complementary in shape and charge. To be honest, I don't know why the answer sheet do and charge. Usually it's complementary in shape and charge to the recognition sequences or recognition sequence to allow for specific binding. I'll give you 1 mark if you have that. What else do you also say? Besides complementary in shape to the recognition sequence to bind, the active site is also complementary in shape for what purpose? Besides binding?
To break the hydrogen bonds between.
No, you don't tell me that. I'm talking about the second structure feature of this active site. Chloe, what's the second structure feature of active sites besides for binding? It's also to allow for. What am I trying to cleave here? By the way, between these bases, do I call it phosphodiester bonds or phosphoester bonds? You cut 1 time phosphoester bonds, not diester. So by the way, the enzyme active site also contains what? Oh, enzymes always do what? You learn in camp, enzymes do what? So they have a catalytic site for what? Catalyze what? Of the hydrolysis of phosphoester bonds. I also accept. Okay, but I need to emphasize it's called phosphoester bond, not phosphodiester. So the answer sheet behind highlight the word phosphoester. Another answer I accept is that you contain, let's say, basic contact sequences, because you should know DNA is negatively charged, so you must be positively charged. Okay, so like NH3 plus is basic ma. So that's why you can actually form ionic bonds to attach to DNA. I also accept that. So that's why I use this as a revision material as well. Now next one, let's see, suggest why different number and length of DNA fragments were obtained by each individual. What do you write, Wani?
I said there could be many recognition sequences within one DNA molecule.
Yeah, so different, not many. They ask you different number. So you just tell me different number of recognition sequence. I will accept. Chloe, do you have another answer? Can I also accept? Different individuals have different in DNA sequences. Understand? So take note. Okay, anyway, for 3A. Megan, can you tell me what is going to be your oncogene? Gene S. Why? What do you say gene S? So what do you see about gene S to make you show that it might be that? There's amplification of the gene, means most likely we can form more protein products or excessive protein products. Remember we did that on the board? So you need to elaborate that. If you never elaborate that, I cannot give you the credit. Understand? But I'll give you 1 mark if you say gene S. Myra, how about you? Do you write another gene besides gene S for oncogene? And there's another one that can be accepted, girls. It's actually gene R. If you look at gene R, there's substitution in codon 56. Substitution of codon 56 can actually relate to gain of function mutation. So like I said, gene mutation can also lead to gain of function mutation of a proto-oncogene into oncogene, remember, under your table that I did just now. Maybe this can lead to, you change your exons, then you will have a hyperactive protein or a protein that's resistant to degradation. I will accept either. Next one. Myra, what is your mutated tumor suppressor gene? Gene P, I accept. Why do you say that? May lead to what kind of mutation? You have that. I'll give you again this 1 mark, loss of function mutation. Alright. So, however, there's also another answer I accept, which is what, Megan? Q. Because you only have hypermethylation of a promoter, that means it's lower or lack of expression of the gene, right? Whether that have loss of function mutation, and that will be a tumor suppressor gene. So I accept both. Next one, you two. Notice how this time you have a restriction enzyme, which is the EcoRI, which is actually the restriction site. So we have, again, with reference, explain how the 2 alleles of gene R can be distinguished using jar electrophoresis and detected by probe Y. Can you tell me, Kalantia? This one, different allele, R1 and R2 have different number of restriction site, right? So they say that if I use probe Y as a detection, for this normal allele on your jar electrophoresis, how many DNA fragments will I observe first? 2 DNA fragments. However, which one will actually show you this probe Y to actually detect? So we have 2 DNA fragments. So how many? Okay, how about R1, R2? How many DNA fragments can I form? So R1 can form 2 DNA fragments. R2 only can form 1 DNA fragment after cutting. So again, question is, for this particular question, they say with reference to figure 3.1, must I code data? Yes, so if you never code that, again, 2 fragments of 168 BP and 231 BP, do you code that? No, then I cannot give you the credit. Same thing with the other fragment, which is only 1 fragment of, you add them together, should be actually 399 KB. That is worth 1 mark. So in the answer sheet, do you see B part 1? The last 2 points is worth 1 mark. You must have both in order to gain 1 mark. Then after that, you need to explain. Idea is that you use your overview point technique or the keyword technique for this explain. Overview point. So overview point is first one is that this gene has actually two different equal RI restriction sites. So what does this mean? You will cut to form different size of, DNA fragments of different sizes. So that will be your first overview point. Subsequent point is the elaboration of overview point. So your this gel electrophoresis will separate based on what? Calanthia. So I can detect the two different fragments on the gel electrophoresis. So gel electrophoresis separate based on what? Charge mass ratio. Difference in charge mass ratio. So you must say the longer the DNA fragment, how will it travel? Longer travel further. Shorter the DNA fragment will travel further, then the longer DNA will travel over a shorter distance. Then I can give you the second mark. Okay? Clear? Okay, next one. Where is your this heterozygous? Heterozygous, that means you have one mutant allele and one normal allele, right? So total how many DNA bands do I see? 2 DNA bands, because remember they are saying they are using your probe Z. Probe Z only binds to one location now. Okay, so I only have 2 DNA fragments. What's the first DNA fragment size? 399, right? So the first one should be your 399. You must draw at the top. Okay, and then the other band is called what brand? 231, because this is the only location on the DNA fragment which your probe Z will bind to. However, will they be of the same size? That means the width of your band will it be the same or different? It will be the same because heterozygous is one each, one allele each. Understand? So that's why same band width. Okay, the rest of it you can use it as study material. With that, I end off here today, so you can see I went through a lot of topics today. So you can summarize and use this as revision material. Again, please go and revise on your this practice paper that I given out. I also given out the answers as well, especially for your this essay and especially for your this immune system question. Go and revise on the answers.